Welcome to the first of 2 lectures on the topic of animal immune systems. As our body's main line of defense, the immune system keeps watch against harmful pathogens. As pathogens evolve new strategies, the immune system in turn evolves new responses. The result of this evolutionary back and forth is an elaborate and sophisticated immune system. The immune system is remarkable in its ability, in most cases, to protect us from harmful pathogens. It is evolved defenses that act against a broad range of pathogens and defenses that target specific pathogens. All organisms have mechanisms that protect them against harmful pathogens. The immune system consists of organs, cells, proteins, and even biochemical reactions that are spread throughout the body. Pathogens are organisms and other agents, such as viruses, that cause disease. For a bacterium, pathogens include viruses, called bacteriophages, that infect bacteria. For a fungus, pathogens include bacteria and viruses. For mammals and other vertebrates, pathogens include viruses, bacteria, fungi, single-celled eukaryotes commonly called protists, and worms. Shown here are several examples of human pathogens. The immune system consists of 2 parts that work in different ways and interact with each other to protect against infection. Innate or natural immunity provides protection in a non-specific manner against all kinds of infection. This form of immunity is present in plants, fungi, and animals and is an evolutionarily early form of immunity. Adaptive or acquired immunity is found only in vertebrates and is specific to a given pathogen. This form of immunity remembers past infections. Subsequent encounters with the same pathogen generate a stronger response from the host. In other words, the immune system adapts over time, and immunity is acquired after an initial exposure. Organisms have a physical barrier surrounding them that protects against infection. Examples include the cell wall of bacteria, the bark of trees, the cuticle of leaves, the exoskeleton of insects, the scales of fish, the shells of eggs, and the skin of mammals. At times, this barrier can be reached, and this can lead to infection. In humans, the physical barrier against microorganisms is the skin. It has 2 layers, the outer epidermis and the inner dermis. The epidermis consists of several layers of cells covered with the protein keratin. The dermis consists of connective tissue, hair follicles, blood and lymphatic vessels, and glands. Like the skin, the mucous membranes of the respiratory, gastrointestinal, and genitourinary tracts act as physical barriers. However, they have additional protective mechanisms. Part of the respiratory tract have cilia, hair-like projections that sweep mucus along with trapped particles and microorganisms out of the body. Saliva and tears contain enzymes that help to protect against bacteria. The stomach, with its low pH and enzymes that break down food, is inhospitable to many microorganisms. Non-pathogenic organisms in the skin and mucous membranes, in some cases, provide protection against harmful pathogens by competing with them for food and space. They may also maintain health in other ways, such as by aiding digestion. We live in a mutualistic relationship with these organisms. If a pathogen makes it past the skin and mucous membranes of an animal, there's a second line of defense, white blood cells, also called leukocytes. The many different types of white blood cells arise by differentiation from stem cells in the bone marrow. Just as the skin and mucous membranes are an external defense against pathogens, white blood cells are an internal defense. Phagocytes are immune cells that engulf and destroy foreign cells or particles. The engulfing of a cell or particle by another cell is called phagocytosis. In this process, a phagocyte, in this case a type of white blood cell called a neutrophil, encounters a cell or particle that it recognizes as foreign and binds to it. The phagocyte extends its plasma membrane completely around the cell or particle until it is within a separate compartment called a vesicle. In some cases, the vesicle merges with an organelle in the cell called a lysosome. Enzymes within the lysosome digest the foreign cell or particle. There are three major types of phagocytic cells in the innate immune system. These are macrophages, dendritic cells, and neutrophils. In each case, their name gives clues to their physical characteristics. Macrophages are large cells that patrol the body. Dendritic cells have long cellular projections reminiscent of the dendrites of neurons. These cells are typically part of the natural defenses found in the skin and mucous membranes. Neutrophils are also members of a group of cells called granulocytes because of the presence of granules in their cytoplasm. The granules of neutrophils take up both acidic and basic dyes, so their staining pattern is considered neutral. Neutrophils are very abundant in the blood and are often the first cells to respond to infection. In addition to neutrophils, granulocytes include eosinophils and basophils. These white blood cell types defend against parasitic infections, but also contribute to allergies. Mast cells release histamine, an important contributor to allergic reactions and inflammation. Lastly, but certainly not least, are natural killer cells. These cells do not recognize foreign cells, but instead recognize and kill host cells that are infected by a virus or have become cancerous or otherwise abnormal. Phagocytes attack foreign cells that they encounter, but leave host cells alone. They are able to recognize pathogens by detecting specific surface molecules that are present on pathogens but not on the body's own cells. Toll-like receptors, TLRs, are a family of transmembrane receptors present on phagocytes that recognize and bind to molecules on the surface of microorganisms. As a result, they provide one of the earliest signs that an infection is present. TLRs detect a wide range of microorganisms by recognizing evolutionarily conserved surface molecules shared by many microorganisms. Binding of the TLR to surface molecules on the pathogen is a signal to the phagocyte to engulf and destroy its target. In addition, phagocytes send a message to the rest of the immune system when a foreign molecule binds to the TLR. Phagocytes release chemical messengers called cytokines that recruit other immune cells to the site of injury or infection. Cytokines, like hormones, provide long-distance communication between cells. While there are generally some immune cells already present at the site of tissue injury or infection, many more must be recruited to fight infection successfully. That recruitment happens as part of a process called inflammation. Inflammation is a physiological response of the body to injury that removes the inciting agent if present and begins the healing process. Inflammation is characterized by four classic signs described by their Latin names: rubor, or redness, calor, or heat, dolor, or pain, and tumor, or swelling. The process begins following tissue injury or infection. Certain cells in the tissue, such as dendritic and mast cells, are activated and release cytokines and other chemical messengers. Some of these chemical messengers recruit additional white blood cells to the site of infection or injury. Others make it easier for white blood cells to reach this site quickly. For example, histamine is released by mast cells and basophils. Histamine acts directly on blood vessels to cause vasodilation, increasing blood flow to the site of infection or injury. Vasodilation causes the characteristic redness and heat of inflammation. Histamine also increases the permeability of the blood vessel wall. Fluid leaks out of the blood vessel, carrying white blood cells into the damaged tissue. The increased fluid in the tissue surrounding the blood vessels is visible as swelling. Some chemical messengers also act directly on nerve fibers, causing pain. Phagocytes can move from a blood vessel to the site of infection. This process is called extravasation. The phagocyte travels along the vessel wall in a rolling motion, grabbing weakly onto the wall as glycoproteins on the phagocyte surface bind transiently to proteins on the endothelial cells. Stronger interactions put a brake on this rolling motion and allow the phagocyte to adhere more firmly to the vessel wall. As it nears the site of infection, the phagocyte encounters and binds to cytokines. The phagocyte then changes shape and moves in between cells lining the blood vessel and into the surrounding tissue. Other white blood cells participate in adaptive immunity, and they can be activated by a set of proteins that circulate in the blood. Collectively, these proteins make up the complement system, which is also considered a part of the innate immune system. The complement system consists of more than 25 proteins that circulate in the blood in an inactive form. This system is activated when these proteins bind to molecules specific to microorganisms or to antibodies. Its activation then sets off a biochemical cascade in which the product of one reaction is the enzyme that catalyzes the next and leads to amplification of the response. Activation of the complement system has three effects. The most dramatic is breaking open cells, or lysis. Complement proteins form a membrane attack complex, or MAC, that makes holes in bacterial cells. The complement system also targets pathogens for phagocytosis by coating bacteria with a protein that phagocytes recognize, a process called opsonization. Activation of the complement system leads to cell signaling in which the production of active proteins attract other components of the immune system. Complement deficiencies are a form of immunodeficiency. They include any disease in which part of the immune system does not function properly. We'll pick up the remainder of our discussion regarding animal immune systems next week.